All right, cell structures. These are the goodies inside of the cell. You will see this diagram frequently as we go through. This particular cell is an animal cell. We know it's an animal cell because it's circular in shape. Plant cells, we'll do in green, are more boxy. Plant and animal cells have almost the same structures. So we will look at those structures first, and then at the end it will show you the plant cells that are, or the plant structures that are specific to plant cells. All right, so as we go through, you'll notice that the structure that we're talking about will be boxed in, so that way you can have the visual. The plasma membrane is the barrier between the cell, the cytoplasm, and the environment outside the cell. Do you remember what I called it earlier this week? It was like our, what? Yeah, very good. This is kind of like the skin of the cell. It protects the cell, it helps support it, it controls what goes in and out of the cell, and it's able to do that because it is what we call semi-permeable. It has the ability to allow only certain molecules in or out. It keeps the good stuff in and the bad stuff out, and it allows like oxygen to come in, but we need to get rid of our CO2, so the CO2 is allowed out. Does that make sense? Prefix semi meaning what? Not half, but
it don't like permit to all of this stuff.
Does it allow everything in and out?
No.
No, just partially, partial or some of. So we are still working with the plasma membrane. The plasma membrane of a cell is what is referred to as a phospholipid bilayer. Phospho indicating that it is made up of with phosphorus molecules. It is a lipid, indicated by the word lipid. And bilayer, what does the prefix bi mean? Two. So how many layers are there? 2 layers. Very good. So you can see that in this diagram here at the bottom. If you split it right down the middle here, you can see that there is a top layer and an inside layer, or an inside and an outside. These phospholipids are arranged to have a head region with 2 tails coming off of it. The head region is referred to as hydrophilic. Any guesses to what hydrophilic means? Water. Hydro means water, so it has something to do with water. Look, hydro, loving, water loving. So then if you're hydrophobic, yeah, water fearing. Good. So the tails, if you look on this blown up diagram, the tails would be hydrophobic. Good. So the tails are hydrophobic. Notice with the orientation of these 2 layers, the tails they go tail to tail, right? The 2 layers. And that's to protect those tails from touching anything watery, because they don't like the water. So they automatically arrange themselves. These molecules automatically arrange themselves so that the tails point towards each other. And then the heads, which like the water, are on the outside of each of the layers. Does that make sense? Okay, so then the next thing to notice in this diagram are these little green things. These little green things would then indicate molecules either entering or leaving the cell. So from the plasma membrane, we can have a couple different structures that come off of it. The first is the flagella. The flagella is a whip-like tail, so it's a little bit longer. And what would the flagella do? What is the function of the flagella? Marta?
Like
Yep. In what is it? Yeah, it helps with movement. There can be multiple flagella. Can be, doesn't have to be. There doesn't even have to be one, but you're not limited to only one. Cilia are more hair-like structures. Again, they will help with movement. And there will be large quantities. A large quantity. All right, moving on. New organelle or new structure. This is the cytoplasm. This includes everything inside of the plasma membrane. This is like the catch-all term. It is not very specific. It's like saying school, not Miss Wolf's classroom. Does that make sense? It's very general. Cytosol is the watery substance that holds everything. It makes it float, kind of buoyant. So it's a watery substance. It will help suspend the organelles so that way they're not crashing into one another. It kind of gives them a home base. It also will push against the plasma membrane, so it's pushing out, so it makes the cell a little bit stronger, and it allows it to keep its shape. And there will also be biochemical reactions happening in that cytosol and in the cytoplasm. The cytoskeleton, this is unique to animal cells. This is a crisscrossing structure in the cytoplasm, which consists of thread-like filaments and tubules. It acts like a skeleton, hence the name cytoskeleton. It helps the cell maintain its shape, and it will also hold organelles in place. Anytime you see the prefix cyto, you should think the word cell. The nucleus. This is the cell's control center. This will be the largest organelle. It contains most of the cell's DNA, and it controls which proteins the cell makes. This is the boss. It is the instruction center. Within the nucleus is a very dense area called the nucleolus. And the nucleolus is the ultimate control center. Does that make sense? Okay. We don't need many more details than that. The mitochondria, this provides the cell with power. So the mitochondria's function is to make energy. ATP, or adenosine triphosphate, is an energy-carrying molecule that almost all cells use for energy. This is what is produced during cellular respiration. So there's this theory called the endosymbiotic theory that states that mitochondria, these little organelles, were once free-living organisms. That means they weren't inside anything. They got to live all on their own, and they had their own DNA since they were free-living. And so what scientists think happened is that there was an ancient prokaryote that infected the larger prokaryotes. So the smaller one was probably the mitochondria, as we know it today, and it decided to infect a host cell. And well, then instead of actually infecting it, it kind of just all worked out. The big cell was happy, the little mitochondria was happy. They both benefited, and so it evolved into this endosymbiotic relationship. So the mitochondria just started to live inside the cell, and now because it didn't need some of the properties of free-living organisms anymore, it lost those characteristics over time. The endoplasmic reticulum, we abbreviate that as the ER, and there are two types. They both help make and transport either proteins or lipids. The rough ER is studded with ribosomes, so it looks rough and bumpy. This is a place for the ribosomes to sit, and the ribosomes then, their job is to make proteins. So ribosomes will either be found on the rough ER, or they can be free floating. And where would they be free floating at? In what structure or what substance? The cytoplasm, and more specifically, the cytosol. Very good. The smooth ER makes lipids and stores other substances. So the smooth ER does not have ribosomes on the outside of it, so it looks smooth. And the way I remember it makes lipids, lipids are the fats and the oils, and they're slippery substances. Smooth and slippery. Does that make sense? Okay. The Golgi apparatus. There is only one Golgi apparatus in a cell, and this processes proteins and prepares them for use either inside or outside of the cell. By processing proteins, what I mean is it is the finishing touches. It is the final place to edit these proteins to make sure there are no mistakes in them, because your cells are constantly making proteins. Sometimes they make mistakes. So if the Golgi apparatus says, oh yeah, no, this one's not functioning properly, it will send it to be broken down and not used. If it is functioning properly, it'll say, all right, you need to go here in the cell to do your job. And so it receives proteins from the rough ER and packages them and sends them to their destinations. It will also do the same thing for lipids. So what you'll notice is there is an order to this in your cell. So when you're building your model cells, you should have this order. So you have your nucleus. Your nucleus contains the instructions for making proteins, correct? Okay. So those instructions then have to leave the nucleus, and they will head to the rough ER. And the rough ER, those instructions will find a ribosome. In the ribosome, it will make the protein. Then the protein will make it to the Golgi apparatus, and then the Golgi apparatus will send it wherever it needs to go. So your order is nucleus, rough ER, ribosome, Golgi apparatus, and then released. Does that make sense? Yes? Yes. So it starts at the nucleus. Then it will go to the rough ER. And on the rough ER, it will find a ribosome. From the ribosome, it goes to the Golgi. And then from the Golgi, it will be released either into the cell or sent out. All right, so vesicles and vacuoles, they have similar functions, and that is to store and transport materials. Vesicles, however, are just smaller. In a plant cells have a massive central vacuole. And that is unique to a plant cell. That massive central vacuole stores water and perhaps other nutrients, but it primarily holds water. Lysosomes are enzymes to break down foreign matter and dead cells. I remember that because Lysol wipes disinfect and break things down. And then peroxisomes use oxygen to break down poison. And the OX in peroxisomes matches up with the OX in oxygen. Oh, yep. All right, so plant cells. These are special structures just in plant cells. They have a cell wall. This is a rigid layer. It's mostly made out of cellulose, and that surrounds the plasma membrane. So you're going to have the cell wall first, and then you'll have the plasma membrane. They'll probably actually be closer. So the blue is the plasma membrane. And then the green is the cell wall. So the cell wall will help support and protect the cell, and it contains tiny, tiny holes in it to allow certain things in or out. Remember, what did we call that? The ability to allow certain things in and out.
The sort of age.
Nope.
That Marta said.
It's the one that Marta said, yes. But what was it? Semi-permeable. Good. Like I said on the previous slide, I believe it was, the central vacuole is a special structure that's just found in plant cells, and it makes up 90% of the cell's volume. So it is like the whole cell. So here would be your central vacuole. It's taking up most of the cell. This again will hold water, enzymes, and salt, and it maintains the plant's shape. If that central vacuole loses too much water, it causes the cell to kind of shrivel in on itself. Does that make sense? Yes? Okay. Other special structures, these are called plastids, and they are unique in that they contain their own DNA. And there are 3 types of plastids: chloroplasts, chromoplasts, and leucoplasts. You know that they're in the plastid group because of plast, okay? You've probably heard of chloroplasts before. These contain the green pigment chlorophyll. Their job is to capture light energy from the sun. Chloroplasts are predominantly found in leaf cells. Chromoplasts would then be any other color. So like a red rose produces a red pigment to make their petals red. So that red pigment is produced by chromoplasts. Leucoplasts then store starches, and they will make some small molecules like amino acids. All right, so here is your diagram of a plant cell. Notice how it's a little bit more boxy on the outside. And that, again, is due to that cell wall, keeping things rigid. Your chloroplasts are found in the cytoplasm again. Here's that big central vacuole. Otherwise, your structures are about the same as a plant cell. And then we can organize cells. If we have a single cell, it only contains one cell. A biofilm is a thin layer of cells. Colonies would then be, usually they're circular in shape. These would be circular groupings of cells. And then we could have all the way up to multicellular organisms. So generally speaking, if we start at single cell and work our way down, we are increasing the quantity of cells. So this right here is your example of a single cell, usually just a bacteria. Your biofilm is up here. Where would you find a biofilm? You experience a biofilm regularly. Not your dead skin cells, nope.
Your eyes or your teeth.
Your dirty teeth. When your teeth feel fuzzy because you haven't brushed them in a while, that would be a biofilm of bacteria eating the junk left on your teeth from your food. Yes, that's gross. Here's an example of colonies of bacteria or colonies of cells, and then obviously a multicellular organism.